Hi, everybody. Welcome to the immune system. Today, we're going to finish up Unit 2 by talking about how your body fights off anything that's foreign. So, as an overview, immunity is going to refer to your body's ability to protect itself from self, non self, viruses, bacteria, fungi, or any other type of disease causing entity. Your immune system consists of lymphoid tissues, immune cells, extracellular and intracellular chemicals, and they're going to be used to coordinate and execute your immune system functions. There's two key features to immunity. One of them is specificity, the other one is memory. So, specificity, what that means is the body's ability to recognize self from non self, and then memory is going to allow the immune system to mount a faster and greater response on an antigen that has previously um, been in the body. So, three main types of functions in the immune system. Here's number one. So, one is recognition and removal of abnormal self cells. Here, what happens is abnormal cells occur when growth and development goes wrong. So, like in diseases that we talk about as cancer, right? So, cancerous cells is basically cells that are dividing rapidly and they have mutated. In this particular case, cancers are detected and destroyed before they get out of hand. So, you have a healthy cell, no binding, no killing, because this is the T cell. T cells kill. T cell in this case is going to bind to and kill the infected cell, which is a good thing because this is the mutated one. In this case, the T cell can bind to the cancerous cell and kill the cancerous cell. It is a weaker binding though because it's a modified self cell. Modified just meaning it mutated. Main function number two is that your immune system is going to remove dead or damaged cells. So we have macrophages that are going to patrol the body and gobble up anything old, dead, or dying. So if you guys remember back from the blood, we talked about white blood cells. So a multipotent stem cell, right, the hematopoietic stem cell, can develop into a T cell precursor, which then moves to the thymus gland. The thymus gland is going to help develop the cell. When it develops the cell, it can turn into what we'll call a macrophage. The macrophage is the patrolling, we'll call it like officer, right? It's like the police of the body. It patrols around and looks for anything old, dead, or dying and gobbles it up. And then number three, the third main function is it protects the body from disease causing invaders. So these, if you guys remember, are the different types of white blood cells. Basophils and mast cells are going to be the granular. Then you've got neutrophil, eosinophil, and monocytes are going to be the agranular. Monocytes differentiate into the macrophages. And then you also have lymphocytes as well. So what happens here is microorganisms, so like bacteria, viruses, fungi, protozoans, parasites, they can all invade your body and cause an illness of some sort to your body. Right? Symptoms are going to differ. So almost any exogenous molecule or cell can elicit an immune response. So like pollen can cause an immune system response, although pollen isn't really going to harm your body. It's just still a foreign invader that your body just doesn't have the ability to identify as non-lethal. Another one is chemical, or there's lots of them, and they all trigger a response and are antigenic. Sometimes your immune system fails to perform its normal functions, and then what happens is pathologies would occur. Um, this is the antigenic. So cancer cells will express the protein. Sorry, back up real quick. Cancer cells will express proteins that are antigenic to the immune system. So basically, all that's happening is this is the cancerous cell, this is the T cell, it binds and is going to kill it through apoptosis. It does so basically by creating these little pores in the plasma membrane, and then it's going to allow water to go in. And you guys know tonicity is a big factor for cell function. So if you have holes in the plasma membrane, basically stuff is going to go in until it's going to burst and die. This is just a different image of it. This is the cytotoxic T cell, and this is the cancerous cell. Apoptosis, the removal of dead or damaged cells. Okay, so actually I was jumping ahead again. So the three main categories of immune system pathologies are: number one would be an incorrect response. Oops. Incorrect response, meaning the immune system attacks itself. So the non-self system fails and the autoimmune disease would occur. So that's the exact definition of autoimmune diseases. Oops, sorry, you guys. There we go. Autoimmune disease is that your body hasn't been able to identify self from non-self, and it's killing off the good cells. So some common autoimmune diseases, Graves' disease. Graves' disease is also known as hyperthyroidism. In this case, the TSH, or the thyroid stimulating hormone receptor on the thyroid cell, you have antibodies that are produced against it and could cause something like goiters. Insulin dependent diabetes mellitus. I'm sure you guys know about diabetes, right? Type 2 diabetes. The pancreatic beta cells antigens, they have antibodies produced against it. Multiple sclerosis has to do with the myelin of your CNS. So myelin, again, is really important because it helps conduction rates, energy expenditure, and it helps with the cell-to-cell signaling. Myasthenia gravis is where acetylcholine receptors are going to be affected at the motor end plate. And we know that acetylcholine is super important because acetylcholine is going to bind and then basically jumpstart the contraction. So it would affect muscle movement. Rheumatoid arthritis is the collagen, most of the time in joints. Systemic lupus is intracellular nucleic acid protein complexes. And then last one is the Guillain-Barré syndrome, the myelin of your peripheral nervous system. So just note, multiple sclerosis is myelin of the CNS, whereas Guillain-Barré is myelin of the PNS. So different nervous systems there. Number two would be an overactive response. So allergies are conditions where the response is out of proportion to the threat posed by the antigen. The systemic effects, though, can be life-threatening. So basically, this is like pollen, an ant bite, even a bee sting. So these are things that are entering different antigens into your body. So we'll call it a foreign antigen. However, the pollen or the ant bite or the bee sting, they're not interjecting antigens to your body that are actually fatal to your body. However, the allergic reaction that your body develops in order to get rid of that non-life-threatening antigen can be very harmful and in some cases lethal. So for example, you're allergic to bees and you get stung by a bee. Now the bee is not putting in an antigen into your body that can kill you. However, anaphylaxis shock will definitely do so very quick. So anaphylaxis is basically all of your systems start to shut down. The reason your body does this so quick and so fast is because your body is perceiving this allergen as fatal. So it's trying to ensure that it's getting rid of or stopping the allergen from getting too far into your body, even though it's non-lethal. So ways that it can stop that, you can shut down your cardiovascular system. You can close off the respiratory system. Like those are two very fast ways to stop things like an allergen from circulating in your body. But you guys know that cutting off your cardiovascular system or shutting down your respiratory system, those are going to be fatal much faster than a lot of other things will. And again, that allergen was never going to kill you in the first place. And then the third immune system pathology would be a lack of response. Actually, I can go through a little bit more on inflammation. Oops, wrong way. So in this case, you have the initial contact with the allergen, then you have the plasma cells that are developed, the mast cells that develop from there, right? They differentiate. What happens upon subsequent contact with that allergen is you're going to have more and more and more allergic responses because your body's trying to fight off that particular allergen. So, the first time you interact with, let's say, pollen that you're allergic to, it's like maybe you have a runny nose. Then the second time you have a runny nose and itchy eyes. Then the third time you have a runny nose, itchy eyes, and your nose starts to get kind of plugged up. So like further and further exposures, the worse your allergic response is going to be because your body again is perceiving it as fatal. Here is a case where you have um, neutrophils are entering the bloodstream from the bone marrow. You have what we call margination, which so they're going to get through. Diapedesis is cell walking, so the cells come through. And in this case, there was like a nail or rust or something like that put into the body. The inflammatory chemicals are going to diffuse from the inflamed site and they're going to act as chemotaxis agents. So you have what's called positive chemotaxis there, and it's going to kill anything foreign that's entering there. This is just differentiation down here, which we've kind of already talked about. But again, there's lots of different types of pathogens that can enter your body. Membrane attack complexes we will talk about at the end of this system, I believe. But basically what they do is they create pores in pathogens. So in the plasma membrane, if you can create a pore in there, what's going to happen is the outside content, which is mainly water and ions, right? So the ECF is going to enter into the ICF. And when that happens, the cell is going to swell and eventually lyse. You lose your structure, then they lose the function. So the lysis of the cell will cause it to die. Okay, last one here is lack of response. So immunodeficiency diseases arise when a component of the system fails to work properly. Primary immunodeficiencies are from genetic inheritance, but you also have acquired immunodeficiencies that are going to be the result of an infection or a side effect of either drug or radiation therapy. Immune response pathways. So immune response pathways, this is the basic pattern of immune response. That is innate defenses when they start first and are reinforced by more specific acquired responses. So there's specific and non-specific immunity, and they're going to be interconnected. So cooperation and communication of the two are going to be really helpful to help fight against any antigen. We're going to talk about three main examples. So number one is bacterial invasions that cause inflammation. Non-specific, actually, let's go back. No, we're good. Okay, so non-specific disease mechanisms are going to be designed to keep bacteria from entering the ECF. So like your skin, mucous membranes, tears, saliva, acid production, these are all designed to keep bacteria out. However, when those barricades fail, bacteria can reach the ECF and usually trigger a non-specific inflammatory reaction that activates a lot of cells to get rid of the invader. So if bacteria enters the lymph, infection fighting will take place on the lymph nodes as well, which is sometimes when you get sick, your lymph nodes get kind of swollen and it becomes hard to maybe move your head in certain ways, or just your lymph nodes start to become more sore for your entire body. In this case, inflammation is characterized by cardinal signs of redness, swelling, heat, pain, as well as joint immobilization. So it's harder to move. So in addition to inflammation, your lymphocytes are going to be attracted to the infected area to produce specific antibodies for that particular type of bacteria. So in this case, so you have leukocytes that are in the blood. They respond to chemical attractants that get released by pathogens. So you've got a tear in the skin, right? The skin was one of those barriers that we talked about earlier. The barricade failed, or the barrier failed. There's a opening in the skin, so a cut, and now pathogens or bacteria can enter into the body. So the leukocytes are going to respond and go through and try to interact now with the pathogens because the chemical attractants have basically called them over. The leukocytes are going to squeeze through the cells of the capillary wall as they follow the chemical signal to where they are most concentrated. That's called positive chemotaxis. Positive because they're going to the area of infection. And then step 3 down here is within the damaged tissue. Neutrophils are going to release chemicals that break the pathogens apart. Monocytes then are going to differentiate into macrophages. Those macrophages then are going to pick up anything old, dead, or dying and phagocytize it and get rid of any cellular debris to basically clean up that area. The idea here, you guys, is that we keep the pathogens out of the bloodstream by having the white blood cells come out rather than the pathogen going in. If you don't know, bacterial infections of your blood is called sepsis. Sepsis or septicemia is very, very, very bad because your blood circulates throughout your entire body. Once you have a bacteria in your bloodstream, it can be traveled to anywhere. It can travel to anywhere and lodge anywhere, and it could be fatal within a couple of hours. Oops. Okay. So the next part of this is bacterial invasions. I don't know that we're going to talk about pyrogens just yet, so I'm going to skip the pyrogens for right now. Engulfment, opsonization. Okay, so bacterial invasion sets off a lot of interrelated reactions. Many of them include the same substances or chemicals to get you to the same end response, which is healthy cells. So getting rid of a bacterial infection, reaction 1 could be activity of what's called a complement system. A complement system, we're going to go through at length at the end of this, but basically what happens is it looks something like this. The components of the bacterial cell wall activate a complement system and they fragment. The fragments of the chemical complement system are chemical signals that we'll call chemotaxis. They attract the leukocytes to the site of infection, and then they act as what we'll call opsonins. Opsonins are an antibody or other substance that binds to a foreign microorganism or cell. This huge thing is the foreign cell. When they do so, they make them more susceptible to phagocytes. So again, I'm going to go over this ad nauseum later on, but what they do, opsonins basically tag this foreign cell. So the idea, you guys, is that your body has to be able to recognize a foreign cell. The only way that it can recognize a foreign cell is if it can identify, this is me, this is not me. So the opsonin, by attaching itself to the cell wall, is basically creating this like highlighter effect all over this foreign cell. And when your body can pick up the highlighter, right, because highlighter is bright, it's an analogy, guys. It's much easier for then your body to identify this as non-self and then go in and kill it. So it attracts the leukocytes, that would be called chemotaxis. Then opsonization takes place and enhances the phagocytosis to occur. Some fragments are going to trigger what we'll call degranulation, so breaking stuff down. Degranulation of mast cells and basophils. The chemicals from these cells are chemotaxic agents, and they're also vasodilators. So for example, histamine is a vasodilator that increases your capillary permeability, and it helps the leukocytes to get to the injured area faster. Vasodilation will increase your blood flow and allow plasma proteins to escape as well, which is why when you have a site of infection, it gets red, hot, and swollen around that tissue. The complement system then is going to end with the formation of the membrane attack complex, which again we talked about as creating those pores. Proteins then insert into the bacterial wall and they cause cell lysis or death eventually. Reaction number 2 is other chemicals help to create the inflammatory response. So you have acute phase proteins. These are that could enter the liver. So liver increases the production of opsonins. It's an antiprotease molecule, and C-reactive proteins, which is the opsonin that activates the complement system. So it would help produce more of those things that are going to tag the foreign cells or highlight them. You also have what we call interleukins. Interleukins are going to be cytokines that are secreted by leukocytes. Primarily act on other leukocytes, but they also affect other tissues. So, for example, an interleukin 1 or an IL-1 is going to alter your blood vessel to help your white blood cells enter the space, activates the liver to make those acute phase proteins, and then it also acts as a pyrogen. Okay, now we'll jump back. A pyrogen is going to be a substance typically produced by bacterium that produces a fever when introduced or released into the blood, right? So, and it's going to cause a fever to occur. And then the last one is bradykinin. So bradykinins are plasma proteins. When they're activated, they form bradykinin. It has a vasodilating effect, like histamine, which also activates pain receptors. Bradykinin. Oops. Next one would be the activity of phagocytes. So bacteria without capsules can be immediately ingested by macrophages. Capsules hide some bacteria from macrophages. So the hiding of them obviously makes it much harder for them to see. So the antibodies must first coat the bacteria so they can be recognized by the macrophages. So here we have a pathogen and then an antigen presenting cell. You have what you'll call an MHC, which is a major histocompatibility complex. And then you have a helper T cell. So what you have here is you have these antigens that are going to bind to the antibodies. When the antigens bind to the antibodies, they're going to help them by making them more identifiable. So we have, if antibodies already exist to that particular bacteria, they are going to enhance the innate response by being opsonins. So this is another type of opsonin, and by neutralizing the bacterial toxins. The antigen presenting cell, or the APC, this is the purple structure here, is going to be able to digest the bacteria and present the fragments to a memory cell. So it presents the fragments to the memory cell, and then initiate more antibodies to be produced. If the infection is new or novel, the antigen presenting cell, or the APC, presents fragments to the helper T cell. Then they activate the naive B cells. The B cells then use their interleukins, and the newly educated B cells, or the ones that are starting to pick it up and become plasma cells. Those plasma cells then can make the antibodies against that particular bacteria. Last part here is initiation of repair. So if you injured a blood vessel in the process, the platelet plug and coagulation can be triggered, which is, we've talked about this before in the, I think we have, right, in the blood section, which was the coagulation cascade, which basically what happens here is by the end of this, you're creating your platelet plug using fibrin, and the injured site can become repaired because you have the plug there, and it's going to allow time for the repair using the growth factors and other cytokines. The platelet-derived growth factor, or the PDGF, activates the fibroblasts for your connective tissues to develop. Okay, viral infections are going to require intracellular defenses. So when viruses invade the body, they encounter innate defenses and typically antibodies. This is not a new virus or a mutated virus. It's just it's something that's already been there. And when that happens, they do it just like bacteria do. So once viruses enter host cells, antibody-mediated immunity is no longer effective. Once the pathogen enters a cell, it can no longer be detected by the antibody immune response. So instead, the cell-mediated immunity response must take over in order to kill the infected cell before it allows the virus or bacteria to replicate too fast and spread to different parts of the body. So cell-mediated immunity is needed. Cell-mediated immunity is going to involve T lymphocytes and natural killer cells, or your NK cells, to help recognize infected host cells and then destroy them. The T cells recognize the infected cells by interacting with antigen present on their major histocompatibility complex. So that's the MHC. Before a T cell can do so, it has to activate with an interaction with the antigen presenting cell, or the APC. So let me see. We've jumped a little too far. So this is going to be too far away because this is the whole mechanism. So I'm going to get into, we can stay here. Okay. So again, cell-mediated immunity involves the T, right? The T lymphocytes, or the T cells, and NK cells as well, to help recognize the infected cells. So the T cells, the killer T, are going to recognize the infected cells by interacting with the antigen present on the major histocompatibility complex. So in this case, you have the MHC1. There's two different types. There's an MHC1 and an MHC2. So if necessary, the T cell will also get activated with an antigen presenting cell, or the APC, like we saw earlier with the APC. If not, you can jump directly to here, where you have the T cell and the MHC already bound to each other. From there, you have a cytotoxic T cell. You might also call this a CD8, that binds the cell to display a foreign antigen. So this is the killer T cell. This is the helper T cell. The difference is what they're going to connect to. So the killer T cell is going to bind to the MHC1 and the CD8 receptor. The helper T cell is going to bind to the MHC2 and the CD4 receptor. From there, it's going to bind to the antigen presenting cell, whereas this one binds directly to the target cell. So this is like the step before you get to this one, right? You need the helper cell to get to the killer T. This is just an intermediate to get to the cell, or the T lymphocyte, which is actually going to kill the target cell, which in this case is a pathogen. So again, the cytotoxic T cell is going to bind to the cells that display the antigen fragments on the membrane protein that you'll call the MHC1 specifically, and destroy the host cell. Once the cytotoxic T cell is activated, it will clone itself, producing many other cytotoxic T cells with the correct receptors. Some portion of the cells are active and help destroy infected cells, while others have an inactive memory and are just going to create more cytotoxic T if the infection returns. From there, your T lymphocytes are going to use T cell receptors, and again, they bind with the MHC. MHC proteins are found on every nucleated cell of the body and are used to display antigenic fragments on the extracellular surface of the cell. So there's two different types of MHC molecules, MHC1 and MHC2. MHC1, again, are going to be located on all nucleated human cells. So when viruses and bacteria invade the cell, they're digested and peptide fragments are going to be loaded onto the MHC1. Cytotoxic T can then interact with the MHC1 using the CD8 receptor. MHC2 are going to be located primarily on the APCs. When these immune cells engulf and digest the antigen, they return their fragments to the MHC2. Then the helper cell, or the helper T cell, can interact with the APC on the MHC2 and secrete a cytokine. That cytokine then is going to enhance the immune system response. The cytotoxic T cells, again, display the MHC1 complexes to prevent the reproduction of the intracellular invaders. So that's really important, you guys. So how do they stop? right, further infection. Cytotoxic T by further stopping their reproduction. They're also going to be responsible for cancer cell destruction and also tissue rejection. So while cytotoxic T cells can be good, they also have the ability to kill kill good cells in a bad way. So like, for example, you have a um, donation of some type. So whether it's, I don't know, kidney or bone marrow or whatever, there's a different type of tissue in your body that should still be good. It matched up theoretically, but your body's immune system fights it off, right? The cytotoxic T cells are fighting off that particular organ or transplant, whatever it may be, as if it's foreign, when in reality, it actually matched up with your body, but your body still perceived it as foreign. Cytotoxic T cells use perforin and granzymes. These are enzymes like trypsin and chymotrypsin, and they're going to trigger apoptosis. They also use what we call death ligands to activate an FAS death receptor. Helper T cells, as you see here, do not directly attack the pathogen or the infected cells. However, they're capable of activating both the cytotoxic T cells and B lymphocytes from there. The helper T cells are going to bind to the MHC2, and use the T cell receptor and the CD4. And again, the helper T cells release an interferon gamma to activate macrophages, interleukins, then would activate the lymphocytes, and what we call colony stimulating factors to help enhance leukocyte production. Your body's immune system response to a viral infection or invasion would be as follows. Let me see, helper T. Okay, to a virus. So to a virus, this is going to be the assumption in this example is that the antibodies to the virus already exist in your body. So step number one, this is going to be broken down into a couple different parts. There we go. Step number one is when the virus invades the host, a pre-existing antibody coats the viral particles, which is going to be opsonization, to make them better targets for the APC, like macrophages. The antibodies can bind to the virus and prevent it from entering into the target cell. The antibodies are no longer effective once inside the host cell. So getting a little bit bigger, right? Allergen is going to be ingested. And oh, wait, this is an allergen. I'm on the wrong one. Hold up. Oh, this is it. Okay, so this is for the virus. Okay, so again, one more time. Pre-existing antibodies are going to be released, and they're going to coat that pathogen, which is opsonization. And number two would be the macrophages present the viral fragments or the antigens on the MHC2, which is that right there, the MHC2, and their membranes. Macrophages then secrete a variety of cytokines to initiate an inflammatory response and activate the natural killer cells and the helper T. So it creates the cytokines and the helper T. They also release an interferon alpha, which are going to activate the host cell production of the antiviral proteins to limit viral replication. The helper T cells then are fully activated when the macrophages present antigens on the MHC2. From there, the helper T cells or the T cell receptor and the CD4 protein interact with the MHC2. The T cells then secrete the cytokines. The cytokines are then going to activate the B lymphocytes and the cytotoxic T cells. So the cytotoxic T cells are here, and then the T lymphocytes are helping for differentiation. The newly activated cytotoxic T cells interact with the infected host cell that present the antigens on their MHC1, and they trigger apoptosis by, again, creating those perforins, or there's holes using perforins and granzymes, which would cause apoptosis to occur. A note here is that natural killer cells can recognize virally infected cells and destroy them as well. What they do is they look for the cells that are lacking the MHC1 complexes, and then they attack in that way. Next part of this is the allergic response. So specific antigens have the ability to trigger allergic responses. Again, an allergy is an inflammatory immune response to a non-pathogenic antigen. So a non-pathogenic antigen is one that's incapable of causing disease. So for example, E. coli. E. coli, it lives naturally in the large intestine, but obviously you don't want to have it in high quantities in different parts of your body. So it's not going to kill you necessarily, but your body does create an allergic response to it. So allergens are antigen that is typically not harmful to the body. If a person is sensitive to the allergen, the body produces an immune response that is with inappropriate ratio. So it's too much of a response for the amount of risk that it's actually presenting to your body. Allergic inflammation or inflammatory responses can be mild to fatal. Sensitivity or hypersensitivity to the immune system response is allergies. Immediate hypersensitivity involves antibodies, and the response occurs within minutes of the exposure of the antigen or the allergen. So, for example, this could be different types of foods. So, some people have peanut allergies. Some people have, I mean, there's so many different types of food allergies. Pick one, especially nowadays, or sensitivities, if you will. Insect venom, so like ants, pollen coming from like a tree. Delayed hypersensitivity involves helper T cells and macrophages that could take several days to develop. Those could include contact allergies to different types of metals, makeup, poison ivy, poison oak. Those are ones that take a while to develop. Allergies have strong genetic components, and you must be exposed to the allergen in order to have that response, the allergic response. So, an immediate hypersensitivity reaction to pollen would occur as follows. Number one is the body would first be exposed to the allergen and go through a sensitization phase. In this case, the allergen gets ingested by the APC or the macrophage. So, the allergen comes into the body and it gets ingested and processed by the APC. Step two, the allergen fragments are presented to the helper T cells. So now it gets broken down, and the antigen presenting cell activates the helper T cells, and now it gets presented. Step three is that the helper T cells activate the B lymphocytes. Their B lymphocytes then are going to differentiate or go one way or the other. They could go one way to be plasma cells, or they could differentiate into memory B and T cells to retain the memory of the exposure to the particular antigen. They do both upon exposure. So step 5 is memory and retaining. Step 6, antibodies. So if you go the route of plasma cells, right, if the B lymphocytes differentiate the plasma cells then are going to secrete the antibodies. Antibodies like an IgE immediately bind to the surface of the mast cell of the basophil. The basophil is this particular lymphocyte. Antibodies bind to the surface of it, and they would then allow the allergen to bind to it and cause the allergic response with cytokines and histamine, which would cause your body to vasodilate, bronchoconstrict, increase vascular permeability, cause inflammation. So all of those typical allergic response symptoms. Upon re-exposure to the same allergen, what would happen is you would trigger a stronger and more rapid response. The allergen binds directly then to the IgE on the mast cells of the basophils that release the histamine, cytokines, and other mediators. If binding to an IgG, not an IgE, an IgG is going to bind the allergen and activate the complement, leading to inflammation and activated T cells. They would do the same thing there as well. The most severe allergic reaction is anaphylaxis. Again, we talked about this already. Anaphylaxis leads to widespread vasodilation, circulatory collapse, and severe bronchoconstriction. Without epinephrine, it can be fatal within 20 minutes. That was a bigger one. Okay. So this is the last part of this lecture, but it is notoriously hard for people to understand. That could be because it's a difficult mechanism to understand, and then accompany that with this super fun picture that's kind of all over the place. There's no one picture in your textbook or one that I've seen that really illustrates what's going on here in a step-by-step process that is consecutive without all these arrows going on, only because this is a very complex pathway. And we're still in, general physiology, or not in immunology or anything like that. So in physiology, I'm going to introduce you to all this stuff, but then in immunology, or in a pathology class, you could take it a step further and actually learn everything that's happening. But going through the overview, it's going to look a little bit messy, so stick with me here. This is what we call the complement cascade. So the complement system is a collection of more than 25 plasma proteins and cell membrane proteins that, when activated, aid in opsonization, so tagging, inflammation, and the production of the membrane attacks complex, or the MAC. The complement proteins mostly come from the liver, are going to be secreted in inactive forms, and have to be activated through several different pathways, which we'll talk about now. So two main ones are going to be the classical and the alternative. The classical pathway is a lectin pathway, and the alternative pathway is going to be used. They're both used to generate a protease that we call a C3 convertase that ultimately will create complement fragments for non-specific immunity. So since the complement system can be extremely damaging to the host cells, there are complement control proteins on the surface of the cells in the blood plasma that prevent the activation of the complement system. So one of them is a C1 inhibitor in the plasma, and the other one's a CD59 in the cell membrane. So what you guys need to learn is the classical pathway and the alternative pathway. So the classical pathway is an antigen antibody complex where you're creating. So let me jump back. We're talking about your body's immune system. Sorry, your body's immune system. So something foreign has gotten into your body, and your body needs to kill that foreign thing before it becomes fatal to your body. Both of these systems, right, the classical pathway and the alternative pathway, are going to kill that foreign thing, but they do so in different ways. So the classical pathway is much more complex. So we'll start there. The classical pathway is this super fun thing. I've broken it down into 10 different steps. You guys can make it however many you want, but the classical and the lectin pathway are, for us, one and the same because they have so many similarities. We're not going to differentiate between the two. So we call it the classical. You can call it the lectin, whatever. It's the same thing. In the classical pathway, step one is a protein is going to create a complex. So this is called a C1 complex. The C1 complex is going to be a group of 3 proteins. So 1, 2, and 3. The group of 3 proteins are going to create a protease. This protease is going to bind directly to the pathogen. So you have the C1 complex that binds directly to the pathogen. This red rectangle is the pathogen. So the C1 complex can bind directly to the pathogen, or it can bind to an IgG or an IgM antibody on the pathogen surface. So in this case, you guys see that the C1 complex is going to be bound to the antibodies, but again, also it can bind directly to the surface. So the C1 complex, again, is the group of proteins. The protease of the C1 complex is going to then cleave. So step one is the C1 complex forms and binds to the pathogen cell. Again, it can bind either directly to the cell membrane or again to the antibodies. That was step two. Step three is that the C1 complex is going to break apart. So the protease of the C1 complex is going to cleave complement proteins into complement proteins C4 and C2. So C1 gets broken down into C4 and C2. What happens then is they group together. So you'll cleave into a C4A and a C4B and a C2A and a C2B. So if you guys can see here, they've cleaved. I think there's some letters that are wrong here. This is how it came. Normally I rewrite this for you guys on the board, but my skills with my cursor are not very good. So I'll see what I can do about that for you guys later on. But just don't forget the C1 complex is going to cleave into C2 and C4 that further divides into the C2A and the C4A and then the C2B and the C2 and C4 are going to divide based on their size, basically. So if you guys can tell, the C4 and C2Bs are going to be bigger than the A's. So they're going to divide into a C4A and a C2A, and then a C2B and a C4B based on size. So from there, that you're going to use what we call a C3 convertase, which is step 5. A C3 convertase takes the complement protein C3 and it is going to convert the C3 and cleave it into a C3A and a C3B. So when you add them all together, right, this step 5 is the C3 convertase takes the complement protein C3 and it cleaves it into a C3A and C3B. You'll have large numbers of the C3B that are going to attach to the plasma surface. So you kind of bring it around here and the C3B will attach to the plasma surface. Step 6 is the C3B attached to the plasma surface acts as opsonization. And again, opsonization, you guys, is the tagging of this particular pathogen as foreign. So it makes it easier. That is going to allow the complement receptors and their membranes to bind to the C3B and initiate phagocytosis. From there, the C4B and the C2B will also bind with the C3B down here, and they're going to create what we'll call the C5. So the C5 convertase will then split into C5A and to C5B, and they're going to move. So the C5A and the C5B are going to cause different things. The C5A is going to go to the host cell, and it's going to cause an inflammatory response. The C5B is going to move to the surface of the plasma membrane. So I skipped over. The inflammatory response could be vascular permeability, chemotaxis, redness, inflammation, all of those. And then the C5B is going to lead to the final step in the complement pathway, where the C5B is going to combine. Here it is. The C5B is going to combine with a C6, C7, C8, and C9. So those are different types of proteins, and they're now going to form the membrane attack complex. The membrane attack complex, again, is going to create the holes or the pores in the pathogen cell lining, and it's going to allow lysis to occur for the cell, which kills the pathogen. Okay, I wasn't super thrilled with the fluidity of that, only because I forgot how fragmented this image is. What I'm going to try to do is draw a better step-by-step process and get rid of all of the excess that you guys don't need just yet. But before I do that, I do want to cover the alternative pathway real quick because it's much faster. So the alternative pathway you can find up here, it's literally an alternate means of creating the C3 convertase. So in this particular pathway, what you have is it occurs on the surface of the pathogen without the presence of specific antibodies. So in this particular part, you have the C3 that's spontaneously going to split into a C3A and a C3B. So some of the C3B attached to the pathogen membrane, in this case, the C3B down here, right, can bind to the plasma protein called factor B and an enzyme that you call factor D, and they split into A and B. The combination of the C3B and the BB, which you'll find here, are going to make an alternative C3 convertase and then merge with a classical pathway to amplify it or make it become bigger. The factor P then stabilizes the C3 convertase enzyme. So amplification happens here when you cleave the C3 into A and B, attaches to the membrane, adds to the BB, and then it's going to amplify it and then eventually cause the convertase, the MAC, and then lysis of the cell. So again, you guys, one more time on the classical pathway, you've got the C1 complex, which is a group of 3 proteins that will attach to the pathogen cell, either by attaching itself directly to the cell membrane or to the antibodies. The C1 complex then is going to cleave into a C2 and a C4. The C2 and C4 cleave further into C2b and C4b, the larger proteins, and then the C4a and the C2a. The C2 and 4b are then going to get added to a C3 convertase, and then they're going to seal themselves to the surface of the pathogen, and they're going to create opsonization or the tagging. From there, you can also attach the C2b to the C3b and the C4 and create the C5. The C5 then is going to cleave into a C5a and a C5b. C5a goes to the host cell for the inflammatory responses. C5b is going to add itself to the other proteins that are C6, C7, C8, and C9, which create the MAC, which open up the pores, which create cell lysis and eventual death. If you guys have any questions on that, please email me and get this solidified, because this is a very big concept for the system and the exam. I hope to talk to you guys soon on Zoom, and I'll see you guys next time.